unless there are some small organic molecules that could absorb the iron 3 and move it away. First possibility. Second possibility, the cells don't oxidize the iron 2 directly. They produce an oxidant that diffuses away from the cells, and this would explain this. So they produce some oxidant problem, we don't know which one. Finally, we can propose that there is oxidation of iron 2 on the cells, and somehow the oxidation is more important close to the cells than away from the cells. Again, right now we have no clue because we know very little about the biochemistry of these organisms. Okay, I'll skip that story. Okay, so that's for these strains that manage to precipitate iron away from the cells. They precipitate the minerals onto organic filaments that are composed of lipopolysaccharides, and somehow there are some gradients that form, gradients of the iron redox state away from the cells. We don't know how they create that, whether iron 3 diffuses away from the cells or if there is a production of oxidant away from the cells. Let's go to the second strain, the one that gets encrusted. This is just first slide showing the cultures. So the medium is here. So this is at the very beginning. We mix iron 2. In that case, we mix iron 2 plus nitrates. If you mix iron 2 plus nitrates at pH 7 in this culture medium, nothing happens for several days. You know, thermodynamically, there should be oxidation of iron 2 into iron 3 because nitrates is more oxidizing, but this is kinetically limited, so nothing happens. If you don't add bacteria, this stays white like this. Now, if you add bacteria, this turns green after a few days, then orange after 6 days. So the microbes really catalyze the oxidation of iron 2 and the reduction of nitrates. If you measure the concentration of iron 2 with time, it decreases down to 0, which means, why that? Because bacteria oxidize iron 2 into iron 3. Iron 3, it's pH 7, so iron 3 precipitate, so you remove iron 2 from the solution. And at the same time, if you measure the bacterial growth, you see that it increases, then it stops. And from this, you could stop your experiment and say, okay, I understand everything. The microbes use the oxidation of iron 2 into iron 3 as for their growth, and that's it. Okay, of course we are microscopists, and our primary goal was just to look at the sample. How do they look like? This is a transmission electron microscopy. This is a high-angle annular dark field mode. So this is a special mode in the TEM. What is bright is iron. Okay, so bright is iron. And so you can see easily the three cells, and you can see where iron is. There are 3 different types of iron precipitate on that picture. The first type is iron precipitate outside the cells, here. Then you have some precipitate on the cells. You see these spheres on the cells. And finally, you have a constant chrome. There is something around the cells, here, iron around the cells. So there's a precipitate around the cells. The funny thing here, if you measure the thickness of this is around 40 nanometer. So this probably tells you something, or give you some hint. If we section this cell, this is conventional ultramicrotomy, and this is another mode. So what is dark is iron, and here the iron precipitate is here, and we could observe an outer membrane and an inner membrane. So basically the precipitate, they start forming within the periplasm. in that volume between the two membranes. These are gram-negative bacteria. So the precipitate form in the periplasm of these bacteria. We can follow this with time, and we see that the first step is really the formation of that periplasmic precipitate before the precipitation of globules on the cells and outside the cells. So this is the first step. We can do the STIXM, we can look at the polymers, the organic polymers in the systems. Basically, this is not that at that time. Okay, we can have this picture. So you see the proteins are in green, so cells are mostly proteins. There are some extracellular precipitate in blue, and the extracellular polymers, the EPS, in blue, and we have in red the iron. Let's have a close-up on this. This is the iron map, this is the protein map. At the iron map we see 3 cells. The protein map, and I promise this is exactly the same spot. Protein map, 1, 2, 3, 4, maybe 5. 5 cells. How do you explain that? Some cells don't contain iron. This one does not contain iron. Okay, so these 3 cells, they got encrusted by iron, that's why they contain iron. This one did not get encrusted by iron. Now, if you think of this, if you look, so the 3 cells that are encrusted, look at the distribution of the proteins. The proteins are concentrated around the cells, while the one that was not encrusted by iron, the protein distribution is homogeneous in the cell. So that's one thing interesting. If you look at a cell that was not encrusted by iron, proteins are distributed homogeneously. That makes sense. If it is encrusted by iron, and remember, the incrustation starts in the periplasm, the proteins concentrate around the cells. We used to go a little further. We use this time transmission electron microscopy, but a special mode. We use cryo-electron microscopy, and we did for this, to prepare the samples, we used cryo-ultramicrotomy. Cryo-ultramicrotomy consists in freezing the sample under high pressure. So basically, you don't have to dehydrate the sample. You keep it in the solution, you freeze this, and then you cut the sample under liquid nitrogen temperature. So it's a bit tricky, but it's possible. And then you look at the sample with very few electrons under the TEM in a sample order where you have liquid nitrogen temperature. And this is necessary if you want to preserve the molecules or the organics. So this is how it looks like now, the cells. Just quickly, this is a cell that is cultured without iron. So you can recognize here the cell wall, and you see the outer membrane, the inner membrane, the peptidoglycan, and so on. So this is a really nice way to image the cells. Now, if we add iron, the first step is the precipitation. What you see here is dense particle here. The precipitation of iron starts on the inner membrane of the cells. So it starts on the inner membrane. Then it grows, so what is dark here is the iron. Then it grows within the periplasm as patches of iron precipitate. So it started on the inner membrane, then grows in the periplasm, make patches in the periplasm, and eventually it invades the whole periplasm. So this cell is completely encrusted within the periplasm, and the precipitate are here. If you look closer to this cell wall here, to the periplasm, the encrusted periplasm, this is how it looks like. What is dark is iron rich. But you see that it's not homogeneously dark. There are some globules, some bright globules, and they measure around 8 nanometers. Now, of course, by TEM we don't know what they are, those globules. We know that they are not composed of iron, because they would be, otherwise they would be dark, but that's it. Now if you compare this with this, remember? Encrusted cells, they have They are rich in proteins around the cells. One explanation, these bright globules are proteins. So, if you accept that, the story is that when you add iron to these cells, they oxidize the iron 2 into iron 3. Iron 3 precipitate on the inner membrane within the periplasm. The cells are losing the proteins somehow. Maybe they are also producing some stress protein, but those proteins are lost from the cells. Within the cells, there's not much proteins left, but those proteins get trapped in the precipitates forming in the periplasm. And that's how you end up with this kind of object. Okay, last thing. This is so the story here is that somehow these cells they oxidize iron 2 into iron 3 in the periplasm. Actually, we started looking at outside the cells. Remember, I told you that there is some iron outside the cells as well. And actually, when we change the condition of cultures, I won't dwell on this, it doesn't matter, but in phosphate-poor medium, this is the kind of extracellular precipitate that form. They are nice hexagons, and those phases, they are crystallized, so we can do transmission electron microscopy on them, and we can give the name of the minerals. And the name, it's not a really nice name, it's green rust. And we know what are these green rust. They are minerals composed of both iron 2 and iron 3. They are hydroxides. They have some carbonates within and some water molecules. What it tells you is that, you see, there is some iron 3 outside the cells. And this is surprising, because as I told you, iron 3 is really at pH 7 doesn't move a long way. So we don't understand how iron 3 could get formed within the periplasm of the cells and then be transferred outside the cells. It's a weird mechanism. This is just something that we don't understand. Except if we change our mind on these iron oxidizing bacteria. Why do we say they are iron oxidizing bacteria? Because, you know, we add iron 2 plus this bacteria and we see that iron 2 transforms into iron 3. But that doesn't mean that there is a direct mechanism of oxidation of iron 2, that there is a genetic process involved in that. Maybe the iron oxidation comes from somewhere else. And this is an hypothesis. I increasingly believe to this hypothesis, but of course it's not demonstrated. One possibility is that those cells, they are not iron oxidizing bacteria. They are denitrifying bacteria. We know that for sure. And so you feed them with nitrates, you feed them with organics, and you feed them actually with acetate plus nitrates, and you add some iron 2. Maybe the iron 2 is a poison. And so they start denitrifying, but they stop at the nitrite step. The interesting thing is that if you abiotically, if you mix nitrite plus iron 2, in few seconds, iron 2 gets completely oxidized into iron 3. Remember, nitrates and O3 plus iron 2, nothing happens. And O2 plus iron 2, in few seconds, iron 2 gets oxidized. So now think about it. These microbes, maybe they just produce nitrites, NO2. And this NO2 oxidizes iron 2. And this explains why you have iron 3 outside the cells, because the nitrite can diffuse away. It's very soluble, it's very highly diffusive, so it can diffuse away from the cells. You have more iron 3 in the periplasm because this is produced in the periplasm, and so that's where iron 2 get oxidized. Second possibility, there is really a specific iron oxidation in the periplasm of the cell, And then there is some export of iron 3 outside the cells, either involving some organic molecules, or there is some lysis of the cells that release some iron 3. But you see here, the idea, if you think about here, the bacteria don't really use the oxidation of iron 2 into iron 3 as an energy source. That would be the implication of that one. In that one, they might use it. Okay, I can skip these things. So, as you see, really looking at the products, the mineral products formed by these bacteria, and looking at the nanoscales, help us to understand how the system works. Not completely. Of course, now we need to get more into the biochemical details. But I really think that there is a need to of interactions between the mineralogy work and the biochemical work on this kind of systems. So, how do this BioFEN cell oxidize iron extracellularly? As I told you, there might be an export of iron 3 from the periplasm to outside the cells. That's one possibility. The other possibility is that the nitrate-reducing bacteria, they produce nitrites, which diffuse away from the cells and oxidize iron 2 away from the cells. So, with this, I'll end up this talk. I'd like to thank several people. First of all, the European Association of Geochemistry and their Romanian Academy and Institute of Biology for organizing that tour and that talk. Mario from the European Association of Geochemistry, who did a big work to organize the tour. Madeleine and Christina, who organized also my coming to Bucharest. I'd like also to thank all these people who have significantly worked on that. Jennifer, in particular, was a PhD student and did the whole work on the cryo-TEM and BioFEN work. And collaborators from Germany. Of course, the beamline scientists from the synchrotron. Synchrotrons are huge facilities, and they work because there are some people who really devote their time to fix all the tiny details of these huge facilities with tons of computers everywhere. And they can be very good. And Gordon Brown from Stanford, who gave me most of the beam time on the synchrotron. And thank you very much for your attention. So I'll be glad to answer your question if there's some time for that.
Thank you for your very nice talk. I would like to ask about arsene. Maybe you know the paper published in Science, which showed that phosphorus was replaced by arsene. Is it possible? What do you think about this?
Or maybe you should stop the movie, because I will answer a kind of tricky question about work of other people. So the arsenic, yeah, this was the science paper suggesting that arsenic might replace phosphorus into the compounds of the cells, like in the DNA in particular. So I think there have been several replies to that paper, so basically comments, sorry, to that paper. So what I'll say is not very new. There is definitely some controversy on that discovery. From what I could follow, one point was saying that, because, okay, some people said that there was not enough evidence of the fact that arsenic was really into the molecules, and so that some controls were missing. At the same time, I was still quite surprised by the fact that one of the experiments shown by the authors said that if we culture the bacteria, if we remove the phosphorus from the medium, the bacteria don't grow. If we remove the phosphorus but we add the arsenate, bacteria grow, as far as I could understand. From that, I thought that, okay, maybe, yeah, there are definitely missing controls, but that observation by itself is quite interesting. If you remove the phosphorus, they don't grow. If you then add up the arsenate, it grows again, so that might be something. But then I know that there have been some comments about the possibility of having some growth for very few generations with very, very little phosphorus that you might add up. So, yes, I mean, one of the points, in my opinion, one of the difficulty, one part of the evidence was brought by the observation of some correlation between the distribution of arsenic and the cells. Like, you can see concentration of arsenic on the cells. Because they were looking at trace element composition, so they were looking at elements in trace concentration. That's a problem. Somebody else showed, for example, that if you look at the map of iron on the cells, you see that it's also concentrated on the cells. And nobody would say that iron is replacing phosphorus, for example, although it's concentrated on the cells. So again, yeah, there are several observations that have been shown in this paper. There have been several comments. So, as everybody, I stay like I'm waiting. I'm looking forward to the next.
Concerning other things, for example, what could be the extracellular filaments rich in lipids and polysaccharide?
Yes, that's something that we have tried to address. There was some kind of speculative thought initially that we had when we saw this, especially when we saw the gradients, the redox gradients around the cells. Let me show you this. Okay, the word is not around, so that's a good thing. But so when we saw the gradients, the redox gradients, something we thought about at once was what was called by some authors nanowires. Nanowires, like so these things, they were discovered discovered. There's still some debate going on. It's quite a big debate on that. In some condition, seem to produce. So there's a discussion whether these are pili or things like that, but seems to produce some filaments, extracellular filaments, and there might be some conduction of electrons. That's what they call them nanowires. And so what has been proposed by, in particular, the Yuri Gorby's group, is that some microbes might exchange or move electrons away from the cells through these kind of nano-electrical wires. So when we thought these things and we saw some redox gradient, we thought, okay, what about nanowires for these bacteria? So our idea was to find some conditions in which we could produce these filaments and maybe extract them and characterize them by biochemistry and so on. So far we have not been able to do that. So
could be these filaments, origin of these filaments could be lipopolysaccharide from external membrane of bacteria. Because, okay, in periplasm it's very clear, because proteoglycan, peptidoglycan can favorize precipitation of iron inside periplasm. But outside, you mentioned that you measure the iron precipitate 40 nanometers. Okay, the lipopolysaccharide LPS, it's around 20 nanometers. Where is LPS in this? Because you mentioned protein and precipitated iron, but where is lipopolysaccharide in this case?
Yeah, so for the precipitation of iron in the periplasm, it's on the other strain. So we really see the precipitation of iron in the periplasm of the encrusting strain. The filaments, we see them more on the non-encrusting cells, the non-encrusting strain, so that one. So both of them, they are gram-negative bacteria. Definitely there is a lipid or lipopolysaccharide. There might be a lipopolysaccharide layer. We have not been able so far with that tool to get down to that resolution. We have not been able really to evidence it. But so the filaments, definitely, I mean, they might be produced by the cell, maybe, and what we know is that they are composed of lipids and polysaccharides, which doesn't mean that there is no protein. There might be some proteins, but just most of the filaments are composed of lipids and polysaccharides. So, I mean, and then the origin, how it forms and so on, this has to be completely, I mean, we have no clue about that.
One simple question, if I may. That green glass mineral you were talking about, is it by any chance modulated or modulated mineral? Do you have a structure for it?
Yes, so there have been a lot of work done on a lot. There have been some work done by other groups on green rust. So these are layered hydroxy salts. So you have a sheath of iron octahedral, sorry. And so there is a structure, and there are actually several structures. Some are substituted by chlorine, some are substituted by carbonates, some are substituted by phosphate or even sulfates. I mean, when I write here carbonate, there are other types of green rust where you have phosphate or sulfate or chlorine. And they definitely, for each of them, some structure has been proposed in the literature. You can find it. And that's how we relied on these structures to infer the electron diffraction pattern that we find here. So yeah, there has been some work. Some people are able to produce this phase abiotically. For example, you mix iron 2 with nitrites, and you get, in some condition, depending on the supersaturation, you can get to there.
You were talking about those vesicles outside the cells. I do not know how to name them. Do you by any chance have some high-resolution transmission electron micrograph through them, so we can see a structure or something?
So, not here. Not here. And the thing is, sorry. No, it's before that. So far, our group, we have been only looking at looking at low magnification to these vesicles. Problem is that we have is that, as you see the cell wall of these vesicles here or anywhere, the cell walls are mineralized, so they are composed of iron precipitates. So somehow it's difficult in that case to observe the organics. The organics is completely hidden by the minerals. Although I showed you some cryo-TEM pictures showing both the minerals and the organics, but the cryo-TEM, we have not been able to do it so far on that samples. So we don't have any high spatial resolution on these ones. Maybe the question is, could we see the organics, the membranes in the vesicles? I think we would need to look at non-mineralized vesicles. It would be easier to see the membranes. There is some membrane and so on. But there are now several papers, or I don't know, I hesitate between many and several papers in the literature dealing with this, what has been called outer membrane vesicles. I think some old works, 20-year-old work by the Terry Beveridge team on this outer membrane work has been done. There is an increasing number of these studies showing that gram-negative bacteria, they can really form these bulges of the outer membrane that eventually can detach from the cells. The mechanism, the biochemical mechanism of this, how you get to separate a lipid, a piece of the membrane from the rest of the membrane, it's still under scrutiny. I think it's not resolved yet. But yeah, that's what we are basing our interpretation on.